So I'm going to go, I'm going to talk first, and then Katrina will follow up. This is an outline of what we're going to talk about, some real basic background about evolution. We'll talk about evolution and HIV evolution at 2 different scales, and then we'll go into some host viral interactions, and then some, a few minutes about questions in the field that we think are important and are outstanding. So first, for evolution. So I wanted to go through evolution at a real basic level for this explainer, just to make sure we're all on the same page. It's one of my pet peeves that people kind of throw around evolution kind of willy-nilly, especially the media and maybe other people who don't really have the same training. It's not right. I don't mean to be pedantic about it at all. I just want to, when I talk about evolution and I talk about evolutionary studies, I want to make sure we're talking about the same thing and understanding why certain pathogens are great for studying evolution, why evolution is really important for some pathogens, and why it's not for others. And it helps, I think, to just get a really basic understanding, a firm understanding of what evolution is and isn't, and why we can use evolution to study pathogens and potentially, inform public health decisions. So first of all, evolution, is really simple. It's just changing allele frequencies over time. There's no mechanism about it. It's just a pattern, as the term evolution. It's kind of taken on in the common vernacular, like a sense of a process, but essentially evolution does entail some forces that change allele frequencies. So these are just the 4 primary forces: mutation, genetic drift, migration in and out of a population, and selection, or adaptation. And by mutation, I'm generally just talking about neutral mutations, because of course the mutations are what selection acts upon. But then, evolution itself is just change over time, or like a pattern of change over time. These are the processes that cause evolution to appear or to happen. But the most important thing that we all kind of focus on, especially in pathogen and infectious diseases, is selection or adaptation. When we see a headline in the media about so-and-so pathogen is evolving to become worse or something, what they actually mean is adapting. They don't mean, oh, it's experiencing genetic drift and it's going to get worse. Or if they say so-and-so pathogen is mutating, it's going to get worse, they don't mean there are just random neutral mutations occurring. What they mean is it's adapting. And so I think as a field, we, even epidemiologists, and computer scientists and biologists, would do better to continue to clarify to everyone, what are the actual key forces that we're talking about when we're talking about evolution. And to me, that is selection and adaptation. So selection and adaptation actually is really, like, the requirements are quite simple, and these are laid out by Darwin. There has to be variation in the phenotype in a population. Evolution occurs at the population level, not really at the individual level. There has to be correlation between phenotype and fitness. And by fitness generally just means the number of offspring that any individual contributes. With HIV, and we'll get into this a little bit, how we define fitness is a little bit murky. Different scales and different types of fitness we're talking about with HIV. And then last, this phenotype has to be at least partially determined by the genotype. And if it is, then it's partially heritable, and therefore this correlation between phenotype and fitness can be passed on from generation to generation, and adaptation can occur. So evolutionary perspectives can be used to explain a couple things. This is just my opinion, and I would definitely appreciate to hear from others, but this is how I try to think about things from an evolutionary perspective. First, why things are variable or conserve over time. So why do things change? So in other words, the focus is on the particular genotype or phenotype. For HIV, for example, is why is amino acid changing at a certain rate over time, either at the population level across individuals or within an individual infection? Why is it changing? And then second, why things are variable or conserved currently. So what explains existing patterns? The focus on the distribution of variation in a population. These two things are really intertwined, of course, but they can be distinct. You can look at a variation in something in a population. For example, set point viral load for HIV. You can notice any cross-sectional survey of set point and notice the Y variation and ask, why does the variation exist? Why is it not all around 4.5 logs? Why does it span 2 to 7? That is a question about the current distribution, not necessarily about whether or not or how set point viral load is changing. To answer one or either of those questions, you necessarily bring in the other, I guess, but they are different questions and I think equally important. So evolutionary studies of pathogens in general, not just HIV, are common, much more common. I think they're becoming much more common and more understood as important to the field. You can see with a massive push for sequencing for the coronavirus, this is, I think, relatively new. Like I assume Christoph and David, your guys' groups I would hopefully agree. This is a really new response due to a lot of things. I think to the general awareness of the importance of sequencing, and recreating phylogenies, which is based on evolutionary theory and evolutionary ideas. It's kind of grown out of the ease of access to technology, for expense, for cost purposes, and also just the quality of the tools. Also, the things that we've learned from other pathogens, including HIV and Hep C and other highly variable viruses and pathogens, what we've learned from looking at phylogenies and sequencing have kind of led us to realize the importance of this type of questioning, and so it's becoming standard in outbreak analysis and pathogen analysis, which is great. And so we can ask these questions from an evolutionary perspective. We can ask about emergence, spread, of course, PANGEA, the central questions of PANGEA. We can reconstruct transition networks, do molecular epidemiology, do phylogeography. These questions range from historical to current focus questions. Ask about resistance to antivirals, antibiotics, host immune response, vaccines, and of course, pathogenesis and virulence using genomics or modeling, or a combination of the previous three. So these are fairly common approaches, things you can ask with evolutionary perspectives for many pathogens, not all. So now I'm going to move on to evolution and HIV in particular. I apologize that this is probably quite background and background knowledge for, I would assume, all of you, but if it's not, that's great. So I think that HIV is unique among pathogens in how often and how integral evolutionary studies are done, and how integral they are into clinical practice and public health action. I think there's still a lot of room and questions about how well real-time phylogenetics can be used to directly change public health practice. I think there's a lot of potential, and PANGEA is pushing us towards a better use of phylogenetics for real-time action, but historically it's been more of a historical science. But then resistance, screening for resistance, of course, when anyone gets diagnosed initially, at least in the West, you do your pulse sequence, and your ART regimen can be specified based on your resistance mutations. This is pretty novel for pathogens. And why this is, probably a few reasons. In my opinion, first of all, HIV evolves very rapidly. So there's a lot of information that we can use in terms of mutations. We see a lot. How do I put this? Let's just go through the slides. So we see that the emergence of resistance can occur within days or weeks within an individual. This is very important, studying evolution, how this happens is really important for clinical practice. For epidemiology and phylogenetics, mutations accumulate faster than transmissions. This is kind of a throwaway point, but actually quite important, and there's been a lot of work done on this, whether or not a pathogen is amenable for phylogenetic analysis to understand transmission is based on a lot of things, but part of it is how fast the pathogen mutates, how much information there is for the phylogeny to use to discern transmissions. HIV is really great in this way because there is a lot of information accumulated between transmissions, so we can really get fine scale transmission networks recreated. Additionally, there's a lot of data available for study. So this includes Los Alamos, GenBank, now PANGEA, all of these sequences that are available and have been available for decades. Gizaid, and flu, and now the coronavirus are following, I think, in this HIV footsteps, and it's fantastic. But this is, for a long time, HIV was, the only real organism with thousands of sequences available for publicly available analyses. Hep C, Los Alamos had a Hep C database, but it kind of has stopped being updated because funding was lost. But HIV has continued, and it's, as PANGEA is part of, it's an incredible resource for scientists everywhere. And most of the sequences are, at request or minimal on Los Alamos, linked to clinical and epidemiological data. And so there's a lot of data for anybody to use, making HIV almost a model organism. It feels a little bit insensitive to describe HIV as a model organism, but it can be. I think a lot of evolutionary biologists, I know a lot of evolutionary biologists who are trained in evolution who have moved into HIV and then on to more infectious diseases because HIV appeared to be such a great system to study evolution in real time. And I also know a lot of MDs and clinicians who have started out studying HIV and then have become experts in evolutionary biology, because it kind of forces you to understand both, or if you can understand, the public health side, the clinical side, the virological side, and the evolutionary biology side, your studies and your analyses are improved. And of course, everyone is still working on this. Of course, I'm still working on trying to incorporate all these different fields when I study any particular HIV data set. So why, of course, and this is some real background slides, why is evolution an issue when we study HIV? We know the HIV mutation rate is very fast compared to any other viruses, especially humans. This is due to the error-prone nature of reverse transcriptions, the step 4 here in the HIV life cycle. A throwaway fact that I like to tell undergrads is that the amount of genetic diversity present in HIV population in one person untreated is roughly equivalent to the amount of genetic diversity among all mammals. This, of course, just speaks to the fact that there is so much mutation and then so much diversity allows us to understand fine-scale patterns of evolution. This is an early analysis from Jim Mullen's lab, showing patterns of intra-host evolution in one person over the course of infection. This plot on the left shows divergence on the top from the founding virus or the ancestor, and this is pairwise diversity of those sequences sampled at these time points post-infection. You can see the divergence continues to increase and then diversity increases. I think this was mentioned earlier in one of the previous explainers in Manon's talk, that diversity, of course, starts to decline usually after the onset of AIDS symptoms. And this is a phylogeny of those same sequences. And you can see it's kind of like a ladder phylogeny, kind of like a flu interhost phylogeny, showing the effects of selection on the viral population within the host over time. This is a recent figure from Morgan and Bethany's recent paper looking at SARS genetic diversity. And this here on the right is a phylogeny of HIV. And this is a phylogeny of SARS, the SARS coronavirus 2. And I'm sure many of you have seen similar types of figures. And this is just a really stark illustration of the amount of genetic diversity in HIV. It's almost incredible that we are trying to do with the SARS coronavirus the types of molecular epidemiology that we do regularly with HIV, given the limited amount of diversity. And if you are following along with a lot of the recent preprints and papers looking at SARS phylogenies, it's really interesting to see how difficult it has been for scientists and people doing phylogenetics to use the same methods on SARS as they do with HIV, just because there are so many more questions and it's much more difficult to make strong inferences about transmission patterns and sources of infection with such limited diversity. HIV is a really incredible case, and that the patterns can be quite strong at times, and it makes studies of evolution and epidemiology almost relatively easy. Well, I mean, compared to other pathogens. So I'm going to move on to real quickly how HIV evolved at two different scales. So first on the left of the intra-host scale, this is really important in discussing how HIV and the human hosts evolve. I hesitate to say co-evolve because I don't think there's been enough real time to date to track and quantify human evolution in response to HIV. I don't know of any real findings. Maybe somebody does. So what we're talking about is mostly HIV evolution in response to the host, and not the reverse. So HIV does evolve at the intra-host scale within individuals, often at the shorter time scale. For example, within a host emergence of resistance to ART or antibodies. But there's also the inter-host scale, which is very interesting. And by this, we mean the population of virally infected individuals. So even though HIV only exists within individuals, HIV doesn't exist in the interstitial space between individuals. Even though that's true, because of the process of transmission, there must be a type of evolution that is occurring at the human population level that may be distinct from the type of evolution that's occurring at the intra-host level. Okay. Katrina is going to talk about this later. And so I show these two trees again, side by side, as a pretty clear illustration of these two distinct time scales or scales of HIV evolution. This is an inter-host phylogeny, of course, and it looks like a star. It's a kind of just really no apparent phylogenetic patterns that would infer the effect of selection, compared to this intra-host phylogeny, in which this ladder-like branching pattern does reflect the selection for a certain type of variant over time. And this is pretty dramatic and suggests that different forces are acting on HIV at these different scales. This is one way to look at this. This is from a recent paper from Rakai and Katrina's group. These are the evolutionary rates for one part of GAG, P24, at the within and between host scales. So the gray background here indicates the between host rates for subtypes A, C, and D. And each one of these columns is a different individual. And it shows the rates of intra-host evolution are much faster than the rates of inter-host evolution. And so this has been known for a few years now for HIV, and there's been a lot of work trying to figure out what this can tell us about transmission. So I'm going to stop here and let Katrina follow up, and I hope that was at least marginally useful.
Great, thank you, Josh. So, so we can think about different categories of host, kind of virus-host interactions, and what we're going to do is kind of start thinking about transmission. So, what it is that makes, what is it that makes a virus or a viral variant good at transmitting from individual to individual, and then we'll think about disease progression, and then we'll kind of think about kind of viral fitness kind of at a bigger scale. So if we think about transmission between individuals, we might want to ask, like, how, what is it that makes a virus good at transmitting, which can be important for lots of reasons. Probably we might want to think about, for example, vaccines, and if we know what are the characteristics of variants that are transmitted, then we can maybe start to think about how to target vaccines better. So the question is there selection on the virus at the point of transmission? And it is a huge amount of effort has actually been gone into asking this question, and it's still very much unresolved. So it's really not clear whether you know, it's just random, it's just stochastic. No, it's just that virus that was lucky that got transmitted. But then there is some evidence that, you know, there are certain traits of viruses that are good at being transmitted. For example, variants that are protected from host innate immune responses. So it seems to be that during the course of infection, then the virus kind of, you know, loses immunity to host innate immune responses because there's no selection for it. But then when you look at viruses that are successfully transmitted, they tend to have these this immunity. So what are the potential causes for variation in HIV transmission probability? Well, viral load, we know almost certainly, so that, well, we know for sure, you know, the higher viral load, the more likely you are to transmit, the viruses to transmit. There's evidence that viruses that are more similar to the viruses that infected individuals, or that are similar to the viruses which are circulating in populations, are more likely to be transmitted. I'm going to skip that one. Yeah, shorter variable loops. Viruses with R5 tropism seem to be better at being transmitted, although that's slightly controversial. If there are viruses with fewer N-linked glycosylation sites, and N-linked glycosylation is thought to protect the virus from antibody responses, and as I was saying before, resistance to type 1 interferons. There's also plenty of evidence that host genetics are important, and host genetics either protect you from infection altogether, like Delta 32, or that they might protect you from infection from particular types, particular variants. So if you've got one particular HLA type, then you may be protected from viruses with particular CTL, susceptibility to CTLs. We know that there's other host factors that are important in prevention, including circumcision and other such things. Inflammation, co-infection, and so on and so forth. We also, well, one of my kind of pet hypotheses is that that viruses generally tend to lose the ability to transmit during infection. So we're talking about these N-linked glycosylation sites, and we tend to get more of those during infection to protect from the antibody response. We tend to see increased sensitivity to type 1 interferons, and we know that escape from antibodies is costly, at least some escapes to antibodies. We also know that if you develop, if you evolve resistance, for example, to CTL escape mutations, then you're going to be those tend to be costly, so they're going to make it harder to transmit to other individuals where those CTL escape mutations are no longer beneficial. And as I said in the previous slide, we know that population consensus virus, so the virus that's most similar to kind of the virus that's circulating in the general population, is more likely to be transmitted. And then here, faster viral fitness, and I'll talk about that in a little bit. So next we can think about disease progression. So what viral and host factors are linked to variation in infection outcomes? So again, we can think about adaptation to CTL escape and to antibodies. We know that if individuals have CTL escapes and some antibody escapes, they tend to revert after transmission because they're not beneficial in the new host. Sites under selection for immune escape are more diverse at the population level. There's a couple of years ago, some work came out showing that the more infecting variants you have, the more severe infection tends to be. And host variants that protect or make you more vulnerable to severe infection. So for example, we can look at HIV adaptation to the immune response. And we can see that when somebody is infected, we see that adaptation actually can begin really soon after infection, so in very early infection and in acute infection. And we see escape from CTL, so we see escape from neutralizing antibodies. But what we also see is that this selection carries all the way on into chronic infection. So for example, here we're looking at two sites of the genome, one in GAG and one in ENV, and we can see that we repeatedly see CTL escape mutations in individuals in GAG, and we repeatedly see antibody escape mutations in ENV. And this has an effect on diversity at the population scale. So going back to what Josh was saying earlier, so we see that those sites in the genome which are more prone to CTL escape or antibody escape, we also see much more diversity in the between host level, so within sequences at the between host level. And this potentially kind of has implications for kind of phylogenetic analyses, because we generally tend to treat sites as being neutral variable sites, and actually this might be far from the case in HIV. Again, we can see when we're looking, again, this is like kind of linking the within and the between host level, but we see that there's a correlation between the frequency of HLA escape mutations and the frequency of that HLA genotype amongst hosts, amongst individuals. And again, we see that there's a correlation between how diverse a sequence variant is and HLA prevalence again. So we're really seeing how kind of selection at the within host level and that interaction with host genetics is really influencing the diversity of the virus at the population level. And then as I promised, we'll get on to viral fitness and viral load. So, so this is kind of really hard talking about this because Christoph is listening, but this really stems from work that Christoph did over a decade ago now. But if we look at viral load during infection, what we all know is we see the set point viral load during infection, and this set point viral load can be quite different between individuals. But what we also see is that individuals with a high viral load, so towards the right of this graph, they're very infectious, but individuals don't tend to live very long, and so they don't tend to transmit so much of the virus. And again, individuals with a low set point viral load have a low infectiousness, but they live for a long time. But because of their low infectiousness, again, they don't tend to transmit very much. So it's those intermediate viral loads which are optimal for transmission. And lo and behold, if we look at the distribution of set point viral loads in a population, we can see that most people have these intermediate set point viral loads. And so what we can hypothesize is that the virus has evolved, has evolved to generate these intermediate set point viral loads. And then the question is, what are the host and viral factors which are involved in kind of generating this distribution? And actually, really, this is still very much an open question, which takes us on to outstanding questions. For me, I think still one of the big outstanding puzzles, and one which I think is really important because it could help tell us about transmission, is why we're seeing these much faster evolutionary rates within individuals than between individuals, and how much can that tell us about transmission. And just kind of like two hypotheses here I'm going to put forward here, and I wanted to talk to you about them because they're hypotheses that both myself and Josh has put forward. People would probably kind of put me in the germline side of things and Josh on the adapt and revert. But I think these really are definitely not either or, and there's really loads of evidence for both. So if we go to the germline theory, it revolves around the HIV life cycle. So viruses, they infect a host cell and then they integrate into the DNA and cause as proviral DNA. But what we find is that some of these infected cells go into this long-lived resting state, which is the HIV reservoir, which means it kind of almost like fossilizes the viral DNA. And then eventually these cells are reactivated. And so you're going to have a mixture of like highly evolved virus circulating in the population and much less evolved virus circulating in the population. And the hypothesis is that these viruses that have evolved less are better being transmitted. Again, remember that we would expect viruses to get less good at being transmitted the longer the infection has been going on. And really the main support for this, apart from it being biologically plausible, is that when we see this mismatch in evolutionary rates, we see it for synonymous mutations, so ones which we don't expect to be under selection. So the other hypothesis is what we call adapt and revert, and here we see mutations. So we might have this individual here who's infected by a purple mutation, and then adapts to the green host with a green mutation and loses the purple mutation. That individual gets transmitted, we get more adaptation, more reversion, and so on and so forth. So the virus is busy evolving within individuals, but actually when you accumulate that over multiple transmissions, actually the virus hasn't done much evolution at all. So again, that could create this mismatch in evolutionary rates. And really disentangling this is really quite important, because when we're wanting to try and, for example, think about what variants are transmitted for vaccines, then we really want to know where those variants are coming from. So this is just really some kind of really general kind of unanswered questions for the field. So what drives evolution during infection? Which variants are transmitted between individuals and why? Can we predict how vaccines will fare? Will this be different for preventative vaccines, for therapeutic vaccines? And would a better understanding of evolution improve our phylogenetic models if we can include those mechanisms into our phylogenetic models? And with that, thank Josh and thank all of you for listening.